day, everyone. It's a pleasure to be on this platform today and to be able to give a talk on sperm evolution and sperm competition in tropical African birds. And this is a part of what I've been doing as research over the years. And of course, I'm not the only one who is doing all this, but with a number of researchers in Europe and as well in Africa here. And of course, you can see that Omotoriogun C and Jan Leifert are very strong collaborators of this project, and they are also my mentors in this research line. So having said that, I will take you through a journey of my interest in sperm morphology studies, and which is also in a couple of years to come, we'll be translating to looking at some other aspects of sperm ejaculates. But today, I would like to give a very specific introduction that narrows to what I'm going to be sharing with you with respect to results and then the discussion. But if you come to realize that the production of offspring is one of the ways in which life is sustained in nature, and that to be able to do this, there must be a union between the gametes, the male and the female gametes. And then you realize that sometimes, and most of the times, you have sexual selection having to have its role in the acquisition of partners. And then the process of that, you have that behavioral traits, morphological traits, and physiological traits have become elaborated across the animal kingdom. And this is just because the fight to acquire partners. And then more specifically, you then have male-male competition because the males would have to compete for females or access to females in order to ensure their fitness. And then in so doing, you then have that there is variance in copulation that cuts across males. But then the females on their part are also very choosy because they want a particular male, the one they find to be of quality and which they want to serve their offspring. So these gymnastics keep going on, and the females keep making their choice and translating to females having to mate with multiple males within a given reproductive cycle. And then you have this mechanism that is called the precopulatory mechanism that help to shape and determine which males eventually have access. But while a particular male tend to have access to females, you have other mechanisms that males that are deprived also want to ensure that they get a fitness. And in so doing, they invest in their gametes, while other males will invest in exaggeration, ornaments to attract females. Some other males will rather invest in gametes. And in this case, it can be the traits of the sperm that they produce. And then if eventually females are able to mate with multiple males, and then you have cases of sperm competition, and where also you have that sperm selections occurs at the post-copulatory level, that's within the female. And these two factors also translates to what you call variance in fertilization. And so these mechanisms are the ones occurring within the females. I'll talk more on sperm competition and sperm selection later. But these factors factors have become, sexual selection has become a prerequisite for the evolution of sperm morphology. And what we have today is that sperm morphology varies enormously across the animal kingdom. And just to give you a quick span of sperm length that you have across the animal kingdom is you have the variation ranging from 7 micrometer in the Omotoriogun species, that's the wasps, and then you add to about 58,290 micrometer in Drosophila biforca. So you can immediately see the variation that occurs in sperm morphology across the animal kingdom. And to be able to assess this variation, a recent paper has also been able to put together data from over 4,000 species, unique species, in 27 animal phyla, describing sperm traits in invertebrate as well as vertebrates. And you can see they describe the head length, describe the midpiece length, flagellum, total sperm length, as well as other type of sperm that exist within the animal kingdom. Now, the factors driving this variation in sperm morphology across the animal kingdom are not fully understood, but over the years in literature, you've had that the environment in which fertilization occurs, as well as the species phylogenetic history and post-copulatory sexual selective foxes of sperm competition and sperm selection has also played a very significant role in driving this variation that you have in sperm morphology across the animal kingdom. So you see more of publications trying to explain these factors and how they have been able to play this influence and how sperm evolve over time across the animal kingdom. But also, it's been investigated that where you have high level of sperm competition, what does it translate to? So making connection between the variation in, that's intermate variation in sperm length, and how this is used to explain the level of extra-pair paternity that you have in birds. And of course, there is a negative relationship that has been observed between EPY, extra-pair young, and also sperm and length variation, as coefficient of variation between males. So how does this, what is the meaning of this? This is to show that if you have a given population where you have high sperm competition, so the tendency is that sperm competition will help to reduce the variation in sperm length that you have in that population, and meaning that you would be having an optimal sperm length that is efficient and, of course, effective in fertilization of eggs. And this is more like stabilizing selection acting on sperm length in a given population. Now, having said that, what do we know about sperm morphology, sperm competition, and others in birds within the tropical Africa environment? Very little is known, if not what we have been doing in recent time to describe sperm morphology and birds occurring within the Afrotropical region. But what has been done, this study had only been carried out in the temperate region. And of course, you then have that in general, sperm length had ranged from 40 to 290 micrometer in terms of length. So meaning that most of this data, information that we have, had originated from the temperate environment. So where does birds inhabiting the tropical environment lies, or what would be the range of sperm length for these birds? And now, why do we have to think like that? Of course, because if we look in terms of ecological requirements and adaptation of species in these two environments, of course, we should expect that the prevailing conditions differ to some extent. And adaptation and trait evolution will rely on the ecology and, of course, of the given environment. In this case, the regions that we're looking at. So if that is the case, we then expect that reproductive traits and mating system should then have a significant influence on how trait evolve in these two environments. I mean, these two traits will differ between birds in the temperate environment and those in the tropical environment. And also come to look at the debate that has been over time on life history differences between regions. And of course, this is in literature where you have that there is a difference in mortality and pace of life, in parental investment, and of course, in reproductive rate and clot size between birds in both the temperate and the tropical environment. So given this, we do expect that there will be some differences occurring in sperm morphology and other traits between birds in both temperate and the tropical environment. So in this talk, I would give you the areas where we have worked in, but this is not all, it's just an extract of the entire work we have been doing. So I will describe we'll describe variation in sperm length and components of African sunbirds, then be able to look at intra and intra-specific variation, and then give you look at correlations among sperm traits, and then we'll compare the data that we have from tropical Africa to what is already existing in temperate environments. And we'll use this to test phylogenetic sigma, sperm length, and components across species, looking at both tropical and temperate species. And then we can also test difference between sperm length of both species of birds, of sperm length from species in African temperate species, looking at specific families where we do have that families where we have species occurring in both the temperate and the tropical environments. Now, how have we collected our data? We've been working in Nigeria and Cameroon since 2010, and in Cameroon 2008. The present work is still going on. And then the data we have so far is, for now, we have more than this currently, but this is what I present to you based on the information that I have on the slides. So for tropical species, we have this number of species and family that we have sampled so far. And then for the temperate species, about 139 species have been covered within 34 families. So when we collect these samples, how do we do? We do the collections through cloud massage and preserve as this. So we collect about 0.5 to 3 microliter of sperm samples and preserve in approximately 300 microliter of 5% formaldehyde. We also use the opportunity to collect blood samples because of the barcoding project we are also doing on African birds. And then once we go to the lab, we're able to process the samples, and what we do is to measure the components and the total sperm length of cells that we found in these samples. So this is how we do the measurement. We use the microscope here. This is a setup for the microscope. And then this is also in the lab where we'll process the samples, just to give you a feel of how these samples are being processed in the lab. And then once we've done that, we extract our data. We extract the total sperm length and data for components. And this data is what we then put together and analyze. So far, we estimate sperm competition, and this is how we do the estimate for sperm competition. So we use this as a proxy for sperm competition, given the information I provided earlier with the association between EPY, estrus per young, and the coefficient of variation between males. So, and we are able to estimate using other formulas the level of estrus per young that is possible with a given coefficient of variation between male. Now, once we've done that, we also did the phylogenetic generalized least squares, and this we do to account for the statistical non-independence of data point due to common ancestry or species. So, and these, you know, you can have two species, sister taxa having similar trait value, and of course, when they are distantly related, you could also have that they either have similar or dissimilar trait value. So all of these have to be accounted for, and phylogenetic generalized least squares will help us do that. And then character state, we also have to estimate the ancestral character states, and we do this by reconstructing the character states using file tools and also ape. These are packages that are used interactively with our package. So then we tested phylogenetic signal and the data that we have, and we use the pages lambda, and we also use the Blomberg's K. This is available if you want details of this in our published papers, but for the sake of time, I won't be able to spend so much to describe how we have done these things here. But to give you a quick understanding, the Paget lambda is a likelihood ratio test that determines and whether the estimated the maximum likelihood value of lambda, whether it's differ from 0 or differs from 1. So if it's differed from 0, we are talking of no phylogenetic signal, and if it differed from 1, we're talking about phylogenetic signal. Just like you imagine your phylogenetic tree, you have nodes, you have divergence, speciation events occurring. So for that kind of tree, you have phylogenetic signal. But cases where you have radiation and polytomy, you do not expect any phylogenetic signal under such conditions. So, and we did for Blomberg's K, trying to quantify phylogenetic signal using these two parameters. Okay. Now, what we have so far is that we have analyzed about 128 species in 28 families. And now I just want to give you a quick visualization of the variation that you have in sperm, in total sperm length across species of tropical, Afrotropical songbirds. And you'll see here that the sperm length range from 51 to 212 micrometer, and that the coefficient of variation that within male that we have for this is ranging from 1 to 5.8 coefficient variation. And then we have indicated here on the super families, the Passauroidea, the Musicapoidea, the Ciboidea, and the Coboidea, and trying to look within these super families the families themselves and the species, and then be able to look at what variation you have. So you can see that here, for example, you have the Musicapidae, then you have the Plossoidae, then you have the other families, the Frigilidae and the Nectarinidae as well. They've Pycnonototus, the Pycnonotidae, where you have the bulbos as well. So now, if you look at this, the color you see is to allow you visualize the gradient in terms of sperm length from low to high. And here we have indicated red is low and blue is indicating species with long sperm. As you can see here, that the Musicapidae have the species in this family do have, long sperm compared to the Cysticolidae, which have very short sperm. And of course, that's within the three superfamilies, but we have generally that within the Covoidea, the sperm length are also quite short. But that's if you look at the Cysticolidae as well. Now, in this data, we are able to tell you that the range of sperm length is from 21 micrometer, which is the shortest sperm we observe, and this is in the single cesticular. And of course, the longest was about 212, which is the streaky-headed seed eater. This is Critagra gularis. And now, I also told you that certain clay stands out with some distinct pattern in sperm length, and that I pointed the ones with the blue, the Frigilidae and the Musicapoidae that have long sperm, and of course the Cysticolidae with very short sperm, of course, compared to other families as well. Now, the Plossoidae also have, if you look at the general there, the Euplectes and the Plosseus. Now you discover that the Euplectes have longer sperm compared to the Plosseus. Now there are also intrageneric contrast in sperm length among Afrotropical species. But if you look at here, this is the Frigilidae. You could see the variation that you have in sperm length. But if you come to look at the Estrididae as well, you can find that the Lagostita sanguinodossalis is standing out. That's the rock biofinch is standing out compared to the other Estrididae. So, this is the variation you have within a given family as well. And of course, as I pointed out before, looking at intrageneric contrast in sperm length. But then, if I want you to have a look at what sperm length or shape look like in this species, this is what I'll be able to project to you. It may not be very clear to you. I don't know how you see it from your end, but this is still looking at the superfamilies. And at this time, trying to tell you the range of sperm length that you have across this superfamily. So it's quite obvious that the Covoidea do have very short sperm length. And of course, across the other superfamilies, you have sperm length, a mix of short and long sperm. So what is this telling us? So the Covoidea seems to be at the very base of the tree, and meaning that sperm length, long sperm length would have evolved from the short sperm length. And of course, you can see this superfamilies having long sperm within this group. So this is just to show you, I'll give you a pictorial illustration of the species that are really involved. So the other is what you have the sperm length to be. So M1 here is for African thrush, and this is African thrush. These are look like, and is the same for us. So trying to arrange it to give you this picture. Now, when the Afrotropical species were broken down, as I've earlier explained, this was what we found in terms of the range of sperm length. So, but the variation in sperm length among Afrotropical spermbed matches the pattern that is observed for major clades, that is above the family levels in the passerine, as described by C. 2007, and where we have longer sperm that is above 100 micrometer occurs in passerine sunbeds, while COVID sunbeds have shorter sperm, similar to the sister group of the passerine and the tyranny that we know about or described in this paper. Now, suggesting that long sperm have evolved frequently within each of the families of the passerids from an ancestral state of short sperm, which I described to be the covoidea. Now, in terms of correlation between sperm traits, we have very strong correlation occurring in sperm trait, but in this case, I've just presented to you sperm head length and sperm total length. And this is a correlation that we have controlling for phylogeny and also between sperm mid-piece and total sperm length. So, you know the role of mid-piece in the sperm, as it is a powerhouse. So, most of the time, mid-piece will have to correlate with sperm length. Now, I'll discuss further on that in the next slides. But let me give you a quick insight into the variation in sperm competition, which I've not, is just what we have found from our study. And here described that the range is from 1.3 to 8.2. What does this mean? That sperm competition varies in birds in tropical Africa. And it could range from high sperm competition, moderate sperm competition, or no sperm competition at all. So if we follow the proxy that we use, you will have that the range that we have in terms of EPY, extra pair young, is from 39 to 0. So across 26 species that we're able to recover 10 males for, because to estimate this sperm competition prosy, we need at least 10 above males to be able to do this. And what does this mean? That if you have a 10% of EPY, it therefore means that the coefficient of variation between men should be around 2, and it should be around 3 if you have a 10%, and if you have of 25, it should be around 2.0 coefficient of variation. And now, in our data, we're able to group the coefficient of variation to low, moderate, and high CVBM. So as you can see here, how they were categorized. So this means that those with low CVBM do have low CVBM would mean high level of sperm competition. And why? Those with low CVBM will have high level of sperm competition, and those with high CVBM will have very low level of sperm competition. Now, this is where we compare the variation in sperm length between temperate and Afrotropical sunbeds. And as you can see that in this phylogenetic tree, the bars here, the red bars are for Afrotropical species, while the black colored bars are for temperate species. So we're trying to group species together. So you can, this just to give you a feel of the variation that occurs. If you look at a particular group or family, you can see how these differ between species from Africa and those from the temperate environments. Now, we have described in details sperm competition and sperm length variation in the Pachynotidae as well as in the Nectarinidae. So those papers are already published. But what am I to try to project to you here that if we visualize the variation that you have, and of course this is expected because generally across passerines you do not have this huge variation in sperm head length, but the variation is mostly in the midpiece and in the flagellum, because the midpiece and the flagellum are very significant parts of the sperm. And of course, in some parts you have the tail. The tail do vary depending on the species. So here we visualize the overlap between sperm length of birds in the temperate environment, those from the tropical environment. And what you see here is that at the very extremes, you have the sperm length of birds from the northern temperate region sticking out. And whereas you have, in this range for northern temperate species, the sperm length of Afrotropical species. And so, but then on the extreme, towards the longer sperm, you have more of species from the northern regions dominating this. So, and suggesting that for Afrotropical species, if we compare with those from the northern temperate environments, and of course, we can say that sperm length is short for Afrotropical species compared to what you have in the northern temperate. This being, we have to pull out species from both that belongs to the same family. So we can look at these species within the same family in both regions and know whether there will be any difference in sperm length between the species in the family. And that's what led to us looking at 4 families with species represented in both regions. And here we're looking at the Hirudinidae, the Frigilidae, and the Musicapoidae, and the Todidae. So we also observed that, sperm length appears to be longer in northern temperate species than what you have for Afrotropical species. If you look at this figure, but there's no difference in, after we've done the test, controlling for phylogeny, we didn't find any difference in sperm length between species from this region. So here is just to quickly show you how we tested for sperm length and components, controlling for phylogeny, significant difference in sperm length, sperm components in this region. And of course, we tested for phylogenetics and sperm length for species across regions. So there's more to explain here in terms of the phylogenetic signal. and which I already have presented in the method, and explaining to you that if you have that this value, the lambda value differs from 0, it means that there's a strong significant, phylogenetic signal, and which is what is reflected in the p-values here as indicating that this value of lambda are significantly different from when you have 0. And same applies for the Blum-Best K. So to summarize, we see that there is similar pattern and diversity of sperm size in Afrotropical and Northern Temperate Zones species, and that African species did on average have significantly shorter sperm, but that this does not mean there's a difference. So because what we found, we did not see that difference in sperm length between this region. So differences mainly is caused by the different family representation, because the families that you have in the Northern Temperate region are not the same with what we have in the tropical region. So this difference in family could be producing that effect that we have in sperm lengths while comparing between these regions. So, and of course, we saw that this agrees with strong phylogenetic signal sperm size differentiation, what variation explained by the deeper nodes in the phylogeny. If you recall that I told you the speciation event looking at the tree, so that will indicate the phylogenetic signal compared to when you don't have speciation events and all you have is just kind of radiation. So if you can imagine the Galapagos fitchings, for example, that kind of radiation will not be able to produce a strong phylogenetic signal in sperm length. And of course, shorter sperm length in tropical than those in temperate zone may be due to selection pressures or constraints operating at the early evolutionary history of the group, and not so much by general difference in contemporary selection pressures between tropical and temperate zone species. So this is how we summarize, but there's more to do. And this is because the sample size, data collection is not exhaustive, and we are still working to have more or expand the database. And this is where I like to use this platform to ask for cooperation and collaborations of individuals, research groups, institutions on this project across Africa, that wherever you're working, it will be good if you can help with sample collection, and then for us to expand database of sperm morphology based in Africa, and also to begin to understand some other level of sperm competition in African birds. We'd like to conduct live sperm motility experiments and to assess swimming speed in birds. And of course, to look at divergence in sperm traits between close relatives, and if for populations are occurring at some regions across Africa. And of course, we'll still have the plans to look at sperm genomics, proteomics, and also metabolomics. And this is a huge gap in this area in birds of Africa, which we are hoping to dive into. And of course, alongside, we'll be able to access aberrant sperm in relation to environmental stress, pollution, and inbreeding or disease conditions in birds. So on this note, I'd like to thank you for listening. And I'd like to acknowledge this people and organization. Thank you very much.